bacterial point of view, but remember that your cells do the same thing, right? So, the food that you eat every day is not just sugar, hopefully, right? You do get other sources of nutrition, like from proteins, even fats, okay? And so we're going to see how those get metabolized, right? So, looking at proteins first, there is one step that requires proteases. These are enzymes that break down proteins into amino acids. You're going to be doing an assay that looks exactly for proteases. And then also the second part of this is that the amino acids get deaminated. That is, the amino groups are removed from the amino acids, and then enters a Krebs cycle. So there is a picture for this. This is from your book. So here is a protein. Okay. Now, what do these individual circles represent? Proteins are made up of what? Amino acids, right? So these are actually individual amino acids. So what a protease will do is that it breaks down the protein into its individual amino acids. Right? So these amino acids are linked together to make the protein. It breaks those links, and now you have individual amino acids. And then those amino acids get deaminated. So what happens is that you remove the amino group. So there's another process where the amino groups are removed, and then that generates something that looks kind of like this. Okay, you have this product here, and this is something that can go into the Krebs cycle. Right? So what we're trying to do is we're trying to convert this protein into something that's usable by the central pathway metabolism that we talked about. Because if you get it into the Krebs cycle, then you can use it as a way to make energy. Right? So some of you are aware of this, right? There are these special diets where you rely heavily on protein. Okay, it's actually not very good for you in the long run. But there are these special diets where you rely almost entirely on protein, and in order for you to survive for any period of time, you actually have to use some of that protein to make your energy, because you no longer have enough sugar around. Okay? And so this is the way that your cells can do this. Right? They can actually divert some of that protein in this pathway, and then here to make energy. Okay, so again, there's two steps. There's protease, and then there's deamination. Okay, those are the two steps right here. Okay. All right, so now let's look at another nutrient, fats. Okay. Here's an example of a fat molecule. All right, so this one has a bit more stuff on it. Here it has these three components. Okay. And then it also has this glycerol linkage. Right. So these are the fatty acid components. This is the glycerol component. There's an enzyme that will break down this fat molecule into its individual components. That's called lipase. Right. So again, you're going to do an assay today to look directly for lipase. And so what that lipase will do is it breaks down this fat molecule into these individual components. Okay? So now imagine that you get this molecule broken down into these individual components. You still need to break this down further in order to make it into something that your body can use. And so that's the next process called beta oxidation. So beta oxidation takes these fatty acids and it breaks it down into two carbon segments. So one way to picture this is if you imagine a fatty acid as a chain of carbons, right, like this. Okay, so let's treat it like that, and so forth. What beta oxidation will do is take this molecule and it'll break it down 2 carbons at a time, like that. Right, so you're left with this. Okay? What these 2 carbon segments become is acetyl-CoA. Right, so acetyl-CoA. Now, where does acetyl-CoA go?
Krebs cycle. Oh, we're not done.
Yeah, so which process does acetyl-CoA feed into? Krebs cycle, right? So this goes to the Krebs cycle. Alright, so again, we're trying to put something into the central pathway of metabolism, because we know that the central pathway of metabolism is how we make energy. Okay, so that's what's going to happen there. Right, so that's how we get something, that's how we get energy from fats. Okay? Right, so that's what happens in that process. And you can see it here on this diagram. This shows you, and also shows you some more details, what happens when you break down a fat molecule. Okay? So here they represent the fat molecule just as carbons entirely. Here's the glycerol part. Here are the fatty acid chains. Right? It's called a triglyceride. Some of you may know about that term. Right? This is why we call it a triglyceride, because there's 3 fatty acids that make up the fat molecule. Lipase breaks it down. Here is the glycerol and then the fatty acids. So here we see that. Okay? The glycerol can actually go into glycolysis. The fatty acids, which we've been focusing on, what they undergo is beta oxidation. Okay? Beta oxidation. And what happens in beta oxidation is you break this into 2 carbon segments, which you can see happen right here. Okay? That goes into the Krebs cycle. Right? Now, what they also show you here, which I didn't describe, is you can also get some other molecules out of this. So FADH2, NADH, right? So you can actually get some reducing power from this as well, which can go in the electron transport chain. All right, so in all cases, you can see how this fat molecule can feed into the different parts of the central pathway of metabolism, what we started the chapter with. Okay, so that's the tie-in to what we talked about earlier. All right, any questions about that? Okay, so again, you have to keep in mind, what you have to know are those 2 processes for proteins and fats, right? Proteases, deamination for proteins, lipases, beta oxidation for fats. Okay? And you're going to see some of those processes in lab today. Okay, any questions about this? Okay, again, this all feeds into the central pathway of metabolism that we talked about, so that you can get energy from these processes. Right, just to add a few other things to this, anabolic pathways. So there's another set of pathways we call anabolic pathways. And we talked about this at the beginning. All right, so anabolic. Are you going from big to small, small to big? Small to big, right? So this is also known as biosynthetic pathways, okay? Right? And so anabolic pathways, there's a term that we use to describe many of these pathways. It's called amphibollic. Amphibollic means it can go in either direction. Okay, so it can be either catabolic or anabolic. What that means, so what that means is that, let's say you have a pathway that goes from A to B. If it's amphibolic, it means it can go either from A to B or from B to A. Here is an example from your book. So this is an anabolic pathway where we start with glyceraldehyde 3-phosphate. Start with acetyl-CoA. We use that to make this molecule, which is then used to make glycerol. We start with acetyl-CoA, which is then used to make fatty acids. So remember, starting with small, making something big. This is an anabolic pathway. And then we combine this to make fat. Okay? All right, so this is one way that you can make fat molecules. All right? Now, this is considered an amphibolic pathway because if you go in the opposite direction, right? If you go in the opposite direction, that's what we just talked about with this process. Okay? How we break down fats. Right? So the biosynthetic pathway is exactly, or the anabolic pathway is pretty much the opposite of the catabolic pathway. And so this is an example of what happens in many of these anabolic pathways. It's amphibolic, right? Your body has a pathway to go in one direction and also in the other direction. It's amphibolic, okay? All right. Any questions about that? That's what that term refers to. Okay. All right. Now, another type of pathway. We focused on the central pathway of metabolism for breaking down sugars. And that applies to a lot of different sugars. However, there are other pathways that can be used. Now, in your chapter, you learn about, you'll read about other different pathways. However, the one that I want you to focus on is this one, which is the pentose phosphate pathway, because that utilizes a sugar we haven't talked about that, which are pentoses. So pentoses are 5 carbon sugars, okay? And they can be formed from glucose, right? But the importance of this pathway, so let me first show you the pathway. So here is the pentose phosphate pathway. Now again, you don't have to remember these details, okay? What I want you to keep in mind is just what happens as a consequence of this pathway. Right? But here you start with glucose, you go through these various processes where you get to a point where you end up with a pentose. Here's one, ribose, here, ribose, right? And then you go through another series of reactions, and then in this process, what happens is, one thing that I emphasize in this is that you get some materials that you can use for biosynthesis, right? So here you get things that you can use for anabolic reactions. Here you can get some materials you can use for to make nucleotides. Nucleotides are used to make what? What kind of molecule? DNA, yes. Okay. All right. So, in fact, right. So you may know this on my shirt. It says DNA on it. Okay. That's one thing you can get from nucleotides. That's what nucleotides are used to build. Is DNA. Okay. All right. So that's the point of this is going through this is that you can get products that are used for anabolic reactions such as nucleotides, and you also get amino acids. Okay. So this is an example of a pathway that's used in that anabolic side of the equation. And as you can see, right, I simplified this because you can also get other types of molecules too that are used in other pathways. Okay. Any questions about this? So again, we're not going to focus on the details of this, right? The main part is to know what you can get out of this, which basically is a description here. It's using biosynthetic pathways, especially nucleotides, amino acids. All right, so this also is a lead-in to what I want to talk about next to close this off, which is the Calvin-Benson cycle. Okay. Now, this is a part of an overall process called photosynthesis, and that's what I want to spend a little bit of time talking about with you. All right, photosynthesis. Okay. All right, so far in this class, what kind of microbial agents, what kind of microbes have you learned about that do photosynthesis? What type of microbes? What's a microbe that does photosynthesis? I heard someone say it. Yeah, cyanobacteria, okay. So there are some bacteria that can do it. What else? There's another whole big group that's not a bacteria that can do photosynthesis. Algae. Algae, yes, okay. All right? Okay, so for some microbes this process is important. And also, as a lot of you know, plants do this, okay? All right, so now, I'm just going to give you go into this briefly to give you kind of a overview of this. One thing in microbiology, especially for a class like this, which is health related, right? There's not a lot of photosynthesis that goes on when we look at health aspects of microbiology. So I'm just going to give you some highlights about this. Right? In other classes, though, especially if you go into some of the biochemistry of this, you'll go into a lot more detail about what actually happens. Okay? What photosynthesis involves is you start with carbon dioxide and water, use energy from light to make this molecule, which is what? Glucose, right? It's a sugar. Okay. And is this catabolic or anabolic?
Anabolic.
It's anabolic, right? You're starting with small molecules and you're getting bigger molecules. that's why you need energy, which you get from the light. When we look at photosynthesis, there are two sets of reactions. There's light-dependent reactions and light-independent reactions. The light-dependent reactions, this is where we use light as part of the process. And in this process, what happens is that there's a molecule, chlorophyll, which many of you heard of. Okay. It captures the light energy, and then that energy gets used to raise the energy level or excite electrons. And those electrons, those high energy electrons, are used to make ATP and reducing power. Okay, so again, we're seeing some familiar themes here. Here is a diagram of one type of light-dependent reaction. Okay, so here is chlorophyll, right? And here's another version where you can see chlorophyll in these in this part of the structure. Okay, that chlorophyll takes light energy and excites these electrons, right? And those electrons then move across these molecules, and that's used to make reducing power. In this case, it's called NADPH, right? And also ATP. Now, if you were to step back and look at this, what does this remind you of that we've already talked about? ETC, right? So this is another version of electron transport chain, but this one's used for photosynthesis, right? So conceptually, it's the same idea. You pass electrons along, right, to create a gradient. You can see the gradient being made here, which then drives ATP synthase to make ATP. Okay, all right, but it starts out differently in that we're using light energy to raise energy levels of electrons rather than using reducing power to start with. Okay, all right, so that's what's used to make reducing power and ATP. And from a photosynthesis point of view, the reason why we need this, the reason why we want this, is that this is used as the energy to be able to make the sugar. Okay? And that's what powers the light-independent reactions, where we use carbon dioxide, right, which is one carbon, we use to make sugar or glucose, which has 6 carbons. And again, you don't need to know the details of this process or of this process. Right? I just use this as an illustration to show you what happens. Right? So again, we start with carbon dioxide. That's what we have here. Okay. And then again, because we have ATP and we have NADPH reducing power, that's used to power this biosynthetic pathway to start with these carbon dioxides and to end up making sugar. Okay? All right? Also, you can see a connection to something else we talked about, ribulose bisphosphate. So ribulose, where did that come from? Where did that come from that we talked about earlier?
Pentose.
Yeah, pentose, right? So it actually comes from here. Okay? All right? So that's where that comes from. And that's used as an important component to be able to keep this process going, the Calvin-Benson cycle. Okay? So this is a light independent reaction because you don't need light for this process. You just need the products of that light dependent reaction, which is ATP and NADPH. Okay. Any questions about this?
Okay, no light needed, just the process. Is that what you said?
Just the products.
All right, so we have a few more minutes. I wanted to give you since in this unit and in the next unit, you're going to be delving into this. So first of all, some of you may notice my shirt. What does this shirt mean? It says 2.9% Neanderthal DNA. What does that mean? Okay, here's the back of my shirt. You notice it's similar to this. Does anyone know what this is the name of a company. Does anyone know what this company does?
Chromosomes.
What do you think they do? Has anyone heard of this company? 23andMe? It's a San Francisco company, South San Francisco-based company. They actually have a very important influence in healthcare. Does anyone know what they do?
Genetic engineering.
Yes?
They probably look at your DNA to see if you have likelihood of certain diseases.
Exactly. So this company does personal genomic testing, DNA testing. Does personal DNA testing. So you want to get your DNA tested, you can actually go through this company to do that. Does anyone know how much would it cost to do this?
A lot.
So it actually costs $99.
Oh.
Right? So for those of you in healthcare, that's cheaper than a lot of different tests that you get, right? Okay. And now, so you can get your DNA tested, and they have over 100 different diseases or genetic conditions that they can look at. Okay, so some of you may have heard about in the news. Breast cancer is one of them. Okay, so there are some genes that are connected to breast cancer. There's a famous movie star associated with that.
Angelina Jolie.
Yeah, Angelina Jolie, right? So, okay, so they can test for something like that. Now, first of all, you have to get DNA. So if you want to do this test, what would they use for your DNA?
Blood.
All right, here's the answer. So what do they use for your DNA?
Saliva.
Saliva. Okay, so they actually give you what's called a spit kit. You fill this tube with saliva, okay, and then you send it away, and then there's actually DNA in your saliva from the cheek cells. Right, so there was a lab that you did earlier with cheek cells, right? Right, and a lot of you saw nuclei in those cheek cells. That's where the DNA is, right? And you can get that analyzed by this company. Okay, so this is what the spit kit looks like. So this is what you spit into. Okay, so I actually did this in class. It was actually in this room in front of microbiology classes. So they actually had to film me doing this. So they're, all right, so there was me trying to fill up a tube. They even timed me, all right? So I actually went over the amount of time that 23andMe thought it would take me, okay? And then I even got tweeted, right? And then what you do is you put in this biohazard bag, and then you put in this box, and then they get you your information, right? Okay? So they can find out a lot of things from you. Like, I don't have that cancer gene, that breast cancer gene, right? So that's good. Okay. They also found out that I'm Asian. I bet that surprised you, right? Right. And so, but something else they can determine is this: they can find out what percentage of your DNA is Neanderthal DNA. Now, what are Neanderthals? Do you know what Neanderthals are?
First.
So it's actually an early form of humans, right? So there's more than just so scientifically, what's our classification name?
Homo
You learned about
sapien? Homo sapien.
We're Homo sapiens, okay?
Oh my God, I'm
right?
fine
There's
with that.
actually other species of humans along with us early on, right? So one of them was Homo neanderthalensis, or Neanderthals. Okay, so there's actually another species of humans besides us. We weren't the only ones historically. Okay, but this was like thousands and thousands of years ago, like some 30,000 years ago. Neanderthals basically went extinct, right? However, some of our DNA can be traced back to them. How did that happen?
They found
So
out.
how would some of the Neanderthal DNA get into our DNA? Breeding, yeah. So now it's thought that we actually interbred. Early on, we actually interbred with the Neanderthals. Okay? All right, so now you can actually see that remnant in your DNA. So one of the things you can do from 23andMe is that you can get your DNA test and see how much of your DNA is Neanderthal DNA. And I have 2.9%. Right?
Is that a lot?
So the range is, you can see from back here, it's from 1 to 4%. Right? Says that somewhere. Hashtag. Right? Okay. So average is 2.8% for Asians. It turns out that before humans migrated to Asia, there was interbreeding between between Neanderthals and what became the Asian population. So Asians tend to have a higher amount. Okay, all right, now I have 2.9%.
Wow.
Okay, all right. So one of your assignments, okay, is there's also someone else who went through 23andMe testing. Someone that you know.
Ceballos?
Yeah, Ceballos, right? So she also knows what percent Neanderthal DNA she has, right? And so that's your assignment, is to find out what percent she has, and also to find out whether I have more than she does. That's what I'm trying to find out, and she won't tell me that.
Found the test.
Right? So, okay, but that gives you a little bit of that's one of the fun things you can do with this, and of course you can actually buy a T-shirt, you know. If you want to advertise to the world how much Neanderthal DNA you have. Okay, any questions about that? So actually you are going to be learning about DNA in this unit, and after this there's a biotechnology unit where you're going to talk about DNA testing. Okay, so this is one practical application, and also a lot of healthcare is going this way, where they're going to be looking at your DNA or your patient's DNA to understand how to predict disease and how to treat disease. And you're already seeing examples of that now, especially with breast cancer. Okay? All right? Any questions? All right, so we are going to take a break now. When you get back, we're going to start a new lab, enzymes. All right? And I also want to get you back your